So I'm going to talk about the high-throughput functional screening that we have performed in the HTS facility at ICGEB in order to identify microRNAs which control cardiomyocyte proliferation. So why is this important? As most of you may know, in mammalian cells, cardiomyocyte proliferation stops as soon as the pups are born, and this makes, this is an issue taking into consideration that in most forms of heart disease, a regenerative capacity would be important. So a considerable effort has been invested to develop strategies in order to restore myocardial mass, and like engraftment of donor myocytes or strategies to promote cardiomyogenesis. But these strategies have been very limiting. So we aim at a strategy of promoting proliferation of existing cardiomyocytes. So all of you have heard in this meeting a lot already about microRNAs, so I will not go through this slide in very detail, but I want to remind you that microRNA genes have been identified in humans, about more than 1,000 microRNA genes. So if you take this number into consideration and also the information that each microRNA gene is predicted to regulate hundreds of targets, it has also been predicted that about 1/3 of the human genome, or maybe even more, is regulated by microRNAs. So taking this into consideration, and also the fact that microRNAs are involved in a broad range of biological processes, this is why we decided to see if microRNAs would also be involved in cardiomyocyte proliferation, and if we could use them as an enhancer of this process. So microRNAs have already been involved in the cardiac function and dysfunction, as you can see in this slide. So for example, microRNAs have been involved in processes like fibrosis or hypertrophy, and they have indeed also been involved already in cardiomyocyte proliferation, and this was restricted to two microRNAs that you have already heard about in one talk yesterday, miR-1 and miR-133. But mainly the results which have been shown for these two microRNAs is that they inhibit cell cycle regulators, leading to an inhibition of proliferation of cardiomyocytes, the opposite of what we are interested in. So as I've said, we decided to perform a screen of microRNAs trying to enhance cardiomyocyte proliferation, and this was performed in the high-throughput screening facility at ICGEB, which was recently implemented. And this facility has state-of-the-art instrumentation to perform RNAi screens and libraries to perform both human genome siRNA screens or mouse genome-wide siRNA screens. And as I will show you, there is also libraries of human microRNAs, both mimics and inhibitors. So this is the workflow of the screen we have performed. So we have the microRNA mimics arrayed on 96 well plates. We transfected by a simple reverse transfection protocol the microRNAs in cardiomyocytes isolated from newborn rats. 52 hours later, we had EDU, which is an analog to BRDU, and 72 hours after transfection, so 20 hours after adding the EDU, we fix the cells and we do the fluorescence staining. And for this screen, we have used two different proliferation markers, which were EDU and the Ki-67 staining, a widely used proliferation marker. And we have also stained the cells with alpha-actinin to stain for cardiomyocytes. And then we used automated microscopy analysis to collect the data. And this is how it looks like. So on the top panel, we have the original microscopy image, and in the bottom panel, we have the image analysis. This microscopy was done in cardiomyocytes transfected with our control microRNA, which is C. elegans microRNA, miR-67, which does not share homology with microRNAs in human, mouse, and rat, and which is not predicted to target genes of the human genome. So what you see here is in blue, the nucleus. In green, the cardiomyocytes staining with the alpha-actinin marker, which is a cardiomyocyte marker. And the red labeled nucleus are the ones which have incorporated EDU. So it means that if you now look at the image analysis, if you have a green cardiomyocyte with a yellow nucleus, is a cardiomyocyte which is proliferating. And so we have determined that about 12.5% of the cardiomyocytes in control situation are proliferating. And I should mention that this is based on approximately 3,000 cells analyzed per condition. So these are the results of the screen. In these plots, what we have is the percentage of cardiomyocytes which are positive both for EDU and Ki67 in both of the replicates that we have performed. In the green dots correspond to the control microRNA. So here you can see several dots because in each plate we arrayed several controls. And then what you can immediately see is that we have a large number of microRNAs which increase cardiomyocyte proliferation more than twofold. These are about 200 different microRNAs. And we have about 40 microRNAs which increase proliferation more than threefold. We also have a large number of microRNAs which decrease proliferation. These are about 300 microRNAs, which are these ones here. So this is the same results, but in this graph I have just highlighted two different microRNAs which increase proliferation, so two top hits of the screen, and two microRNAs which decrease proliferation a lot, because I want to show you the data, how it looks like. So these are the two examples of microRNAs which increase the proliferation. If you look at the images, you can immediately see a lot of red nucleus, which in the data analysis corresponds to cardiomyocytes with yellow nucleus. And you can see that in this for these two microRNAs, we have increased proliferation from 12% to about 50%. For the microRNAs which decrease cardiomyocyte proliferation, we have here two examples, and you can see that you cannot find yellow nucleus. So we have a reduction of proliferation to almost nothing. And these are two examples of microRNAs which decrease proliferation without affecting cell viability. So one way or one thing that we looked at to validate the results of our screen was to microRNA families. So microRNA families are defined based on the sequence of the microRNAs, the similarity of sequence of the microRNAs, and specifically of the seed sequence. And the seed sequences are the nucleotides between 2 and 8 of the microRNA, which are essential for the binding to the target messenger RNA. Here I'm showing you the members of the LED7 microRNA family, and as you can see, all of them share the same seed sequence. So we look at several of these families, and what we have seen is common effects for the microRNAs of the same family. For example, miR519 and 302 families both increase proliferation at a significant level above the control. The same is true for LED7 microRNA, which all the LED7 microRNA family members decrease proliferation of the cardiomyocytes. So this is a way to validate the results of our screen. So another way of showing you the results of the screen is to look at cardiomyocytes which are positive for the proliferation markers we have used versus the number of cardiomyocytes in the well, which we can also determine since we did the microscopy-based screen. And as you can see, these are the two microRNAs for which I have already shown you the results, which increase proliferation, which clearly increase the number of cardiomyocytes in the well above the control level, which is this gray dashed line. We have here the two blue dots, which correspond to the microRNAs which do not increase proliferation. So they have the same number of cells in the well as the control. In these images are examples of long-term experiments we have performed. So after the transfection of the microRNA, we have waited for 6 days. And this is how the well of the cardiomyocytes transfected with the control microRNA look like. And this is how the cardiomyocytes look like after transfection with the top hit of the screen. As you can see, the number of cardiomyocytes is much higher here than in the control situation. So we have done the screen using the Ki67 and EDU markers. So Ki67 labels cells which are actively involved in the cell cycle, whereas BRDU labels cells which have undergone DNA synthesis, so S phase. So we wanted to validate the results of the screen using additional markers of proliferation, namely the phosphorylation of histone H3 in the serine 10, which labels cells in late G2 and which are undergoing mitosis, and the localization of AuroraB kinase in the midbodies. So midbodies are structures which are formed when the cell undergoes cytokinesis and which remain a little bit longer after cytokinesis has happened. So these are examples of the images. So again, here we have the control, and here we have a top hit of the screen, a microRNA which increases proliferation. And as you can see, there is a strong increase in the labeling with the phosphostone H3 antibody as compared with the control. These are just representative pictures of the labeling here to show you that this necklace is indeed a necklace of a cardiomyocyte. And this is the quantification that we have done. So you can see here I'm showing you just 6 different microRNAs that we have validated, but we have validated about 40 microRNAs which increase proliferation and about 20 microRNAs which decrease proliferation using this same system. And you can see that these 4 microRNAs we have chosen also increase the labeling with phosphostone H3, and the ones which decrease proliferation, phosphostone H3 labeling is decreased. The other marker that we have used, as I said, is AuroraB midbody localization, and midbodies look like it's shown here. So these are 2 different cardiomyocytes where the cells have just finished to divide, and these green dots or stretches are the structure which is formed when the cells are dividing. And these are 3 different images. So this is a cell which is just dividing. This has divided a little while ago. And again, in these images, you can see the control situation where you can only find cardiomyocyte, which has undergone division, where here you can find several cardiomyocytes which are dividing. And we have also quantified this. And again, we can see that for the microRNAs which increase proliferation, we also see a strong increase in the percentage of cells which are positive for midbody localization, whereas for the microRNAs which decrease proliferation, we see a decrease of midbody localization. And finally, I want to show you some very recent data that we have obtained regarding the effect of the microRNAs in vivo. So what we have done for these experiments is to inject the microRNA directly in the heart of newborn rats at day 0 of the experiment. And after 1 day, we have injected EDU, again, to use as a proliferation marker. And 4 days later, we have collected the hearts. And these that you see here are slices of the injected hearts. So the first panel corresponds to the control microRNA, and the 2 other... I'm just finishing. And the 2 other microRNAs, and the 2 other panels correspond to 2 microRNAs which were hits of our screen. And as you can see in the bottom panel, the green cells are cardiomyocytes or alpha-actinin staining, and the red is cells which are labeled for EDU, so which are proliferating. And as you can see, the number of red necklaces that you see for the 2 microRNAs, it's much higher than in the control situation, showing that indeed these microRNAs that we identified in the in vitro screen can be taken to the in vivo condition. So to summarize what I've shown you that we have performed high-content fluorescent microscopy-based screening in rat neonatal cardiomyocytes in order to identify microRNAs which may promote proliferation. And using different markers, we have been able to show that about 200 different microRNAs are able to efficiently, significantly increase cardiomyocyte proliferation, and also that we are able to identify microRNAs which are able to completely block proliferation. And I should mention that this was the first screen with microRNAs which was done in primary cells. So we are very happy with these results, because we had no idea how microRNAs would regulate proliferation in primary cells. And I've also shown you very recent data showing that selected microRNAs can increase proliferation in vivo, specifically in newborn rat hearts. And we now want to take this to heart disease models to see if they will have a therapeutic effect. And finally, in the acknowledgements. So first of all, I would like to acknowledge my mentor, Mauro Giacca, and all the lab members of the molecular medicine lab, especially Serena Zakina, with whom I'm doing the in vivo experiments, and also Miguel Mano from the HTS facility, with whom I did the screen. And I'd like to thank you for your attention.